recording, and I welcome you, Jaykumar, to come and present.
Thank you, Alina. Thank you, everybody, for joining and spending your precious time with us. Today, we're going to be talking about something very interesting, a radical approach to vaccines. There are two distinctive elements here. One is broad spectrum protection, and the second is open or equitable IP. So broad spectrum protection is also known as non-specific, heterologous, or off-target effects. And in lay terms, we're talking about, instead of the current model of one vaccine for one disease, we're exploring the potential of one vaccine to protect against many diseases and also across variants. And the second, open or equitable IP, this is particularly germane given that vaccines are in large part publicly funded, and they are less effective if not available to the whole world. So the social, economic, legal, and public health implications of such a radical approach would, we submit, indeed be profound. And the need, as we know, is dire. Humanity is bedeviled by a parade of pathogens, epidemics, pandemics, endemic diseases that end lives and crush economies and cause trillions in economic loss. So given that the cost of exploration of some of these vaccines, many of which already exist, is minuscule, and the upside is astronomical, and the chances of success, as we will hear, based on the decades of clinical trials that already firmly establish these broad spectrum effects, and a new understanding of mechanism, given that the chances of success are decent, I personally submit it would arguably be a severe dereliction of duty not to explore this field more aggressively. So with that, I will share my screen. And also we have some folks in the waiting room. One housekeeping note, Alina, Bob is trying to get in, but is having some difficulty. So
All right.
let me...
Thank you.
So as you'll hear today, the science is burgeoning behind this. There are decades of clinical trial evidence, and thank you to the WHO SAGE group for being a pioneer in this area. It's starting now to seep into the public consciousness based on the scientific papers. We now have a cover story from Newsweek, a long profile in the New Yorker, national press coverage in India, and a very widely viewed article in the New York Times. So we're starting to move from a sort of specialized area into something that's more firmly established and understood and recognized. Why are we having this meeting today? We have several desired outcomes. The first is to call for support and additional studies on existing vaccines. Now, I'll go through this very quickly as the speakers will touch on these points. One, we want more deliberate infection or challenge studies. The second is a global flu trial to prepare ourselves for future flu pandemics. The third is to look at existing vaccines against COVID-19 in combination with the new vaccines. They may work very well. They may potentially increase duration and efficacy and address variant evasion. There are a number of studies already on the old vaccines and COVID, and they vary sometimes in results, but they can be reconciled by looking at whether they addressed young healthcare workers or older people, BCG naive populations, and the strain of BCG used. With regard to childhood vaccination studies, there are some new studies that we would like to see done. One is revising the schedule of child vaccinations. So not really a scientific change, just a sort of programmatic change. And that could save potentially 1 million lives, 1 million children a year. The second is looking at BCG revaccination of children who did not develop a scar when receiving BCG, and thus are perhaps less protected. And the third is studies on the relationship between BCG and DTP in terms of tolerizing effects and reducing all-cause mortality. There are interesting studies, and we'd like to do more on BCG for respiratory infections. The Activate-1 study in Greece found a marked decline in all respiratory infections amongst elderly people who receive BCG. We'd like to see more studies in chronic disease, such as type 1 and or type 2 diabetes. We'd like to see more studies on NOPV and OPV and their non-specific effects. And all of these, we'd like to see higher power studies with an aim of possible regulatory approval. The second set of the last three outcomes we'd like today just to set the table, and we'd like this to be a very concrete and impactful meeting, is to explore the development of new vaccines that deploy some of these innate immunity principles that we will discuss today. Taking the old ones and improving on them and working from this new scientific paradigm of innate immunity, which enables this crucial broad spectrum, almost universal protection. And these new vaccines would hopefully be under an open and equitable IP regime. Lastly, in terms of substantive points, we have some policy thoughts. We'd like those people present here and their various groups to consider, to the extent relevant, some policy changes. One is regards BCG for neonates, very newborns. Based on studies, we think that monitoring that, whether the vaccination status at 7 days, that coverage at 7 days would be interesting rather than later, and also monitoring for the presence of a scar. We would like the recommendation to be more specific. Take BCG not because it's a mediocre TB vaccine, but give it to your child because of the non-specific broad spectrum effects. And lastly, there's some good data on the varying efficacy of the various strains of BCG, and we want to make sure that the most efficacious strains are the ones that are used wherever possible. Another policy point is regard to pandemics and epidemics. Our current response to the pandemics was fast and great, but it was funneled down one path, a sort of a framework institutionally, funding, policy, media of novel, proprietary, specific, narrow vaccines. But we submit that the portfolio should be broadened. Let's not place all our eggs in one basket, and also look at these non-specific effects, at the broad spectrum vaccines, and also those under an open IP regime. Let's pursue both. And lastly, in terms of our goals today, we'd like to get some sense of follow-up, and we would love follow-up meetings with WHO and to engage with the SAGE process at WHO, and also to start a process where WHO considers recommendations regarding the points 1 to 3, and also even, we know that WHO is not necessarily a funding body, but it has scientific and public health influence, and perhaps recommendations to the funding bodies that this agenda be pursued would be very helpful. With that, I thank you very much. We have a slew of amazing speakers, and I'll turn it over to Alina to be the MC. Thank you.
Thank you so much, Jay. And I would also like to thank you and thank the rest of the panel. We have Dr. Soumya Swaminathan here, and we are so excited to continue this presentation to her and her team, as well as the whole audience that is here. The next incredible academic that I would like and professor that I would like to take the pleasure in introducing is Dr. and Professor Christine Stabell-Benn. Professor Christine Stabell-Benn is an MD, PhD, and Doctor of Medical Science from the University of Copenhagen. She has worked at the Bandem Health Project in Guinea-Bissau since 1993, and in 2012, she was selected by the Danish National Research Foundation to establish and lead a center of excellence on research for vitamins and vaccines. Since 2013, Dr. Benn is a professor in global health at the University of Southern Denmark. She participates in a large scale of vaccine initiatives with an in-depth network. And through this initiative, she has studied vaccination programs and their implementation in many different African settings. Without further ado, I pass the mic to Christine Stabell-Benn, and thank you for your presentation.
Thank you so much for the kind introduction. I actually think Mihai was going in first and I was going second, but I guess it doesn't really matter to me. So if you're okay with that, Mihai, I will just jump into my presentation and then refer to areas where I know that you will be covering the story in more detail. I'm joining you here from Africa. I hope my internet will allow us to continue this presentation, where I will be talking about repurposing of existing vaccines from an epidemiological perspective, and not least an African perspective, with a focus on translation of research findings into policy. We already discussed this just a week ago at the Optimize 2022 conference in Denmark, where we had the honor of having Dr. Tedros opening our conference, stating that not only do we need new and better vaccines, but we also need to examine how we can use the tried and tested tools in the form of existing vaccines in new ways, including for their off-target or non-specific effects. And in the panel discussion, we discussed with Kate O'Brien, head of the Department of Vaccines at WHO, among others stakeholders, whether we can start using vaccines for their non-specific effects already now, or if not, which studies are needed. I think we reached a consensus regarding several important potential policy changes. And one thing that we felt was ready for policy adaptations was the BCG vaccine against neonatal mortality. It's not only a low-hanging fruit, but actually a fruit already lying and potentially rotting on the ground while we are not taking action on the quite strong research findings that Jaykumar also alluded to. So we have here the WHO review of non-specific effects, which was conducted in 2014 and published in 2016, and it included 5 randomized controlled trials and 9 observational studies, and a combined analysis of these studies yielded that BCG was associated with almost a halving of child mortality, an effect which could clearly not be explained based on the prevention of tuberculosis. Since the publication of the systemic review by WHO, we conducted another trial 3 here on this graph of BCG vaccination to low birth weight children. And all the 3 trials from Guinea-Bissau show uniformly beneficial effects of randomizing children to BCG at birth versus the usual delayed BCG vaccine in the range of a 38% reduction in all-cause mortality in the combined analysis. And this was due to reductions in death from septicemia and respiratory infections. And these findings were recently corroborated by Prentice et al. In Uganda, where they randomized Ugandan neonates to BCG at birth or at 6 weeks and saw almost 30% reduction in the risk of physician-diagnosed non-TB infectious diseases among those who got BCG immediately at birth. And Mihai will be talking about the immunological mechanisms, which can explain very well how this vaccine against tuberculosis could actually have such dramatic impact on the risk of other infections. It is important because neonatal mortality remains high. So while the world has seen a decline in child mortality over the last decades, neonatal mortality hasn't declined to the same extent as also noted by Dr. Tedros in his opening remarks. And BCG is not given at birth. So here is data from Guinea-Bissau, from the rural areas, where you can see that by 1 month of age, it's only or less than 40% of all children who have received the BCG vaccine recommended at birth. This would have been more than 50% if all children had been vaccinated at their health center contacts. The reason why this is not happening is that BCG is only perceived as a pretty mediocre vaccine against tuberculosis. And therefore, there's no emphasis on delivering it at birth. And furthermore, there are vial restrictions because BCG comes in vials of 20 doses, and most health centers are reluctant to open these vials unless there are 10 to 12 children present to be vaccinated. So across Africa, here in Guinea-Bissau, elsewhere, health centers only vaccinate perhaps once a week with BCG, or perhaps even just once a month, leading to huge delays in the BCG vaccination delivery. And it's not only about getting BCG to the child, it's also getting it correctly into the arm of the child. Because here is a summary analysis of all the studies, all of them conducted in Guinea-Bissau. Unfortunately, nobody else looked at this so far. All the studies which have looked among BCG vaccinated children at those who got a scar versus those who didn't develop a scar after vaccination. And you can see in this combined analysis that overall having a scar is associated with almost 40% reduction in all-cause mortality. You can also note here on this graph that the range of children, or the proportion of children who develop a scar after vaccination, varies tremendously from 52% and up to 93%. And this is not because children have inherent different genetics. This is a quite homogeneous Guinean population. This is because the vaccine technique means a lot for whether you develop a scar or not. So we have shown in many studies now that vaccinator is the most important determinant of developing a scar. And this also means that there's a huge potential by improving vaccination technique and focus on vaccination technique and correct delivery on improving the scar rate and thus improving the survival of children. So based on the data that I've presented here and much more data underlying it, I think we have reached a stage where there are some clear and immediate policy changes at the WHO level, which could make a world of a difference. First and foremost, it would be to recommend BCG vaccine, not only as a vaccine against TB, but also a vaccine against neonatal mortality. So currently BCG is recommended at birth by WHO, but though the position paper mentions non-specific effects, it's not part of the justification. So I know from the ground here in Africa, it would make a world of a difference if BCG was actually advocated as a vaccine that could reduce the child's risk of dying in the first month of life. That message would be super important, and it would be even more important, or also important, to accompany that with changes in the vaccine program performance indicators, because currently vaccination programs are only evaluated in terms of the BCG coverage they reach when the child has reached, when the child has reached 12 months of age. So there's no focus on timeliness and quality of the BCG vaccination. That could be changed, and healthcare workers could be incentivized if the performance indicators included also coverage by 7 days of life, and also the proportion of BCG vaccinated children with a BCG scar at the time of DTP3 vaccine, usually around 3-4 months of age. And that assessment at 3-4 months of age could also then subsequently lead to revaccination of children who didn't have a scar. So apart from creating an emphasis on getting a scar, it would also potentially form the basis for other policy changes, which I'll touch upon later in this presentation. Importantly, these two above points should also be accompanied, as Jaykumar alluded to, the introduction of quality indicators for WHO pre-qualified BCG vaccines. As we know, there are considerable variations between the BCG formulation with respect to a lot of factors, including their non-specific effects. But with these immediate and minor changes in the WHO messaging and evaluation of the vaccination program, all the available data suggests that we could make a huge impact and we could reduce neonatal mortality worldwide. I'll move on now to areas where we might not be completely ready for policy changes. We may argue about one or several of these points, but where at least the panel discussion led to a form of consensus that it would be nice to see final large-scale randomized controlled trials to resolve remaining issues in relation to policy change. And these randomized controlled trials, everything suggests, would show that there was basis for making policy changes. The areas I'll touch upon, and there are more, but I've focused here on 5 areas. This is BCG revaccination of scar-negative children, having a live vaccine last vaccination schedule, which prioritizes that children have a live vaccine as their most recent vaccine throughout childhood. It's a focus on OPV and NOPV campaigns to reduce child mortality, not only to eradicate polio. It's BCG against respiratory infections in the elderly and live vaccines as bridge gap vaccines in pandemics. So let's go through them one by one. BCG revaccination of scar-negative children, we already talked about. I have the graph here again. It looks like there is a huge potential for reducing child mortality by having a correctly applied BCG vaccine that results in a scar. But that also means that if a child is BCG vaccinated and doesn't develop a scar, there's still potential for improving its health by giving a correctly applied BCG vaccine. It actually used to be WHO policy to give BCG revaccination of scar-negative children, but that was changed since studies showed that there was no real benefit in terms of improved TB protection of providing a revaccination. But we would propose that a randomized controlled trial with randomization to revaccination or no revaccination of scar-negative children at the time of DTP3 vaccination with overall mortality as an outcome would be likely to show that BCG revaccination, or a policy of giving BCG revaccination to scar-negative children at the time of DTP3, would reduce all-cause mortality by 1/3. The next area is this emphasis on a live vaccine last, and that is actually an outcome of the WHO systematic review, because based on the estimates in that review, Frank Shan, a professor from Australia, calculated that a revised schedule B here on this figure, using the same or existing vaccines but just in a slightly different manner, could prevent 1 million child deaths per year. He proposes also in this paper how this could be verified in randomized controlled trials, randomizing to a second dose of BCG versus placebo with the third dose of DTP3, and also by randomizing in countries that use a DTP booster to DTP booster or no DTP booster. And again, provides the documentation, the calculations in the paper that leads to the estimate that this would reduce or prevent 1 million deaths per year. So here, if the data is to be believed from the WHO systematic review, and there is nothing that has disputed it, we have an enormous potential for making a tremendous impact with the existing vaccines just by using them in a smarter way that optimize both their specific, well-known specific effects and also their non-specific effects. Let me move on to the next area, and this is an area which takes up a lot of our attention for the time being, because we have recently found out that the OPV campaigns that have been provided during the last decades to eradicate poliovirus have been associated with a tremendous reduction in all-cause mortality. When we first started analyzing this data 5 years ago, we saw in Guinea-Bissau that the mortality before versus after versus before the campaigns was dramatically reduced. And this was not just a result of having any campaign, because we didn't see the effects for other campaign types than oral polio vaccine. So we went to collaborators in Burkina Faso, Ghana, and Bangladesh, and we've been able to verify these findings in quite different settings. So this really suggests that OPV campaigns has been one of the major drivers in the decline in child mortality during the last decades. And also worryingly that this decline may be halted and mortality may even paradoxically start to increase again once poliovirus is eradicated and OPV is phased out, currently believed to be in 2026. But there is a new novel OPV which has the same capacity to protect against polio, but without the ability to mutate into polio-derived or to vaccine-derived polioviruses. And it might have the similar non-specific effects as OPV, but it isn't known because it hasn't been investigated for these effects. So it would be really important to conduct randomized trials. And here we would propose a trial mimicking a campaign. So on top of the routine vaccination program, randomizing children to novel OPV versus placebo and following them up for all-cause mortality. And the prediction would be that both these strains would be associated with at least 15 to 20% reduction in all-cause mortality. And this would also mean that we actually would have the possibility to use NOPV as a vaccine to reduce child mortality even after poliovirus has been eradicated. The next area is BCG vaccine against respiratory infections in the elderly. So in small randomized trials from Greece, US, India, Indonesia, and Japan, BCG booster has consistently been associated with lower risk of respiratory infections, including a lower risk of COVID-19 in the elderly, as here illustrated with the Activate-1 trial that Jaykumar also mentioned, where elderly people plus 65 with many morbidities were randomized to BCG or placebo at discharge from hospital and had almost a halving in their risk of new infections in the subsequent 12 months. These are small trials, so we propose, even though they look very encouraging, that a large-scale multi-center trial be conducted to confirm the findings from smaller RCTs. But if they are confirmed, I think we also have a finding here which is directly translational into policy, because this would really be a low-key, low-cost, cheap, safe intervention that could improve the health of elderly, not only in low-income settings, which we've talked most about so far, but also in high-income settings. Finally, I'll talk about live vaccines as bridge gap vaccines during pandemics. We have seen in the trials that have evaluated BCG during COVID-19 that a booster dose of BCG was associated with a reduced risk of COVID-19 in vulnerable groups, whereas it didn't make much of an impact in the people who were already healthy. And this is actually quite understandable when you look at Mihai's immunological data, which shows that it is quite hard to improve much on a winning model. But interestingly, in this meta-analysis we did with Mihai, based on the trials that have been published using BCG versus placebo against COVID-19, there is a suggestion of a 40% reduction in the all-cause mortality of participants who received BCG versus placebo. So I think these results are quite encouraging in terms of potential use of BCG against pandemics, and also MMR and OPV has also shown beneficial effects. And I think Mihai will touch upon how there's actually reason to believe that the effect of BCG might be stronger against another virus type, namely influenza. And this means that we will propose that here in peacetime, while we fortunately have no current influenza pandemic, we should conduct a large-scale multi-center trial on many continents at the same time, testing BCG versus MMR versus, ideally both, versus placebo during a normal influenza season. If we see the anticipated impact of these live vaccines on the normal seasonal influenza, there would be all reason to believe that they would also be effective against a new influenza strain with pandemic potential. So we would already have that in our toolbox should such an influenza strain arrive. So with this, I will conclude my presentation. I've argued that we have findings, scientific findings, which are directly translational into policy in the form of BCG against neonatal mortality. I've also argued that we are on the brink of being able to make policy changes in other very important areas. And I think it's pending on getting that sufficient funding for large consortia go together to conduct the large-scale trials, which could once and for all resolve some of the remaining issues in relation to the 5 points I've mentioned here on this slide and which I went through in my presentation. But I also want to emphasize that this is only the beginning, because there are multiple other possibilities, in my view, for optimizing the effect of vaccination programs by taking into account not only their specific effects, but also the non-specific effects of vaccines. So on that positive note, I want to thank you all for your attention here from Africa.
Thank you so much, Christine. What an incredible presentation. Without further ado, I want to present your colleague and co-panelist, Mihai Netea. Professor Mihai Netea is a full professor of experimental medicine at Radboud University and a leading expert in the field of non-specific effects of vaccines. His main research interests are pattern recognition of fungal pathogens and the induction of antifungal immunity, primary immunodeficiencies in innate immune system, and the study of the memory traits of innate immunity. Professor Netea is known for his breakthrough in the area of trained immunity, a program of innate immune memory and in health and in disease of Science magazine. Netea's research attempts to translate the information obtained through the assessment of human genetic variation in patients into novel diagnostic and therapeutic approaches. Since pioneering his research, Netea has been awarded the WRO Gosling of the Infectious Diseases Society of the Netherlands in 2006, the European Society for Clinical Investigation Awards for Translational Research in 2013, and the NWO Spinoza Prize in 2016. With so many accolades, I am very happy to present Mihai Netea and his presentation, and I transfer the mic to you. Thank you, Mihai.
Thank you very much for the kind presentation and also for inviting me to share with you some of our thoughts today. And I would like to talk today about a couple of things after the great presentation of Christina. I will, in the first part of my talk, I will go a little bit into the immunological mechanisms that are behind the non-specific effects of live vaccines, live attenuated vaccines. Thereafter, I will discuss a little bit about some of our studies in the Netherlands with BCG during the pandemic. And in the end, a couple of slides about what we think would be an approach in the future in terms of developing new vaccines, combining non-specific and adaptive immunity. So I will not go into all the introductory slides through the important epidemiological studies that have been done and which were presented so nicely by Christina, showing how BCG and other vaccines such as OPV, measles-containing vaccines, and so on, have on overall mortality and how do they protect. We were interested to try to understand what is happening, what are the mechanisms behind, and also in the first phase, looking at these epidemiological studies, we wanted to see whether we can reproduce these data in a model of experimental human controlled infection. And for that, of course, we cannot infect the people with dangerous pathogens and so on, but we can use another live attenuated vaccine, which induces a mild viral infection in our case as a model of human infection, and we use for that yellow fever vaccine. Because yellow fever vaccine virus is an attenuated virus, and we can follow it in the circulation with a PCR. We can follow also the inflammatory patterns by measuring biomarkers and so on. And to make a long story short, what we have observed when a group of volunteers received placebo and another group of volunteers received BCG, and 1 month later, everybody received yellow fever vaccine, and we assessed the strength of the inflammation, the strength of the disease, the viremia, the number of particles of the virus in the circulation of these two groups of volunteers, we have observed that the group of the volunteers who received BCG 1 month earlier had significantly less virus particles in their blood. And that was associated also with less inflammation and less side effects of the infection. Now, through these, we showed basically that we can reproduce in a controlled manner the epidemiological data. This did not lead to decreased and deficient antibody titers against yellow fever, because those were perfectly normal 90 days after the yellow fever vaccination. So BCG was able to not to disturb the increase in the antibody concentrations. Now, after observing these things, we asked ourselves, how can we explain these heterologous effects of BCG vaccination? Usually, we explain the long-term effects of vaccines by the induction of immune memory in B cells and T cells, and they take care for specific antibodies, for specific effector T cells. But in case of heterologous protection, we cannot use this explanation because this B and T cell memory is antigen specific, is specific only for the target pathogen. So we turned our attention to innate immune processes, such as cellular populations, such as macrophages, monocytes, neutrophils, NK cells, because they do respond to infections, but they are not antigen specific. And what we have observed in follow-up studies is that purifying different cell populations, and I'm just showing you the data for monocytes, but similar observations we have made with neutrophils and also with NK cell, is that upon isolations of these cells after BCG vaccination, they respond better to stimulation, not only after stimulation with Mycobacterium tuberculosis, which is the specific pathogen, but also after stimulation with Staphylococcus aureus or Candida albicans. And what is the molecular mechanism behind this observation that is mediated by important changes in chromatin architecture? The chromatin, the DNA in our body, is very tightly packaged at the moment that we are healthy without an infection, because we do not need to produce proteins which are necessary for host defense, cytokines, chemokines, and so on, without an infection. However, this conformation is strongly changed during an infection. There are chemical changes in the histones from the chromatin, acetylation, methylation, phosphorylation, and so on, which opens the chromatin to be able to be read and to induce gene transcription. And after elimination of the infection, we always thought that everything goes back completely to normal, and that is true, but with one difference. There are some chemical changes in these histones, in the chromatin, which persist, and they are a sort of bookmarking in the DNA. So the next time the cells encounter an infection, the DNA can be opened much easier, much quicker, more efficient at the right place. And this results actually that after the secondary infection, the enhancement of the immune response is more efficient, and the host defense is better, and the survival is better. So this explains basically why some vaccination, some mild infections as well, can induce an improvement of the immune response after the secondary infection. And this we have observed after several of the live attenuated vaccines. The molecular mechanisms through which this process is regulated are complex and concern both immunological signaling through pattern recognition receptor, changes in the metabolism of the immune cells, and long-term influence on epigenetic enzymes, which allow these changes in the chromatin to persist and induce this increase in the capacity of the cells to respond to infection. These changes take place not only in the circulating cells in the blood, because they are there only for a couple of days, but they are taking place also in bone marrow progenitors of these innate immune cell populations. And by changes in the bone marrow progenitors of immune cells, these effects of the vaccines persist for months and even years. And this explains actually the long-term beneficial effects of BCG and other live attenuated vaccines. Now, based on this concept, we propose a model in which the level of the innate immune responses influence actually the outcome of an infection. For example, if someone has good innate immune responses when they encounter a pathogen that would lead to an inhibition of the growth of the microorganism, for example, of viral infections, low viremia, low systemic inflammation, low systems and survival. However, if somebody has low innate immune responses at home where they don't even know that they have the infection, this will allow the pathogen to grow, to have a high pathogen load, high viremia in the case of a viral infection, which would lead to inefficient systemic inflammation, high severity of the disease, and unfortunately, in some individuals, even death. With this concept in mind, we have performed several clinical randomized trials, and I think that Jaykumar and Christina mentioned our Activate trial performed in elderly with high risk for infections, in which half of them received placebo, half of them received BCG, and we followed them up for 1 year, and we observed 40% less infections in the BCG vaccinated individuals. And it was especially the respiratory tract infections which were lower. With this in mind, we performed the second trial, the Activate 2 trial, which was done during the pandemic. Activate trial was done before the COVID-19 pandemic, but we repeated this revaccination study in Greece during the pandemic, and we followed up the vaccinated and unvaccinated individuals for 6 months, and we observed again that revaccination with BCG in this Greek population resulted in significant protection against COVID-19. With these results, we also performed a follow-up study in the Netherlands in which the population is not vaccinated at birth with BCG. So we were curious to see, we wanted to know whether similar protection can be seen also in a Dutch population which is not vaccinated at birth. And we performed the BCG Prime study in which we vaccinated 6,000 volunteers either with placebo or with BCG. Unfortunately, in this trial, we did not see any difference in the total number of infections. So a similar number of COVID-19 diagnoses were present in the placebo and BCG group. However, despite of this, we have seen very important differences in the way in which the immune response of the infected people reacted to SARS-CoV-2. The individuals who were vaccinated with BCG responded with a better cellular response, so their monocytes and T cells responded better to the stimulation, but also the serological response, the antibody response against SARS-CoV-2. You can see here in the panels in the line C, both IgG, again, S protein, N protein, and RBD were significantly better in the BCG vaccinated individuals who got the COVID-19 vaccine. Based on that, we thought also, would it be possible that there is then not a difference in the total number of infection, but maybe a difference in the severity of the infection? And we looked at the COVID-19 related admissions in our cohort, and we have seen approximately 15% less hospitalizations due to COVID-19 in the BCG group. Interestingly, however, and this has to do probably with differential effects, stronger effects of BCG vaccination for some pathogens compared with others. When we looked at the diagnosis of influenza infection in the BCG versus placebo group, we have observed approximately 50% less diagnosis of influenza. And this is in line with an Activate study, which was done before the COVID-19 pandemic, when the most of the respiratory infections were influenza rather than COVID-19. And this study was also the potentiating effect of the BCG revaccination was also shown in a study done by colleagues from UK and India, in which they have shown that the BCG revaccination improves the response to Covishield, one of the new COVID-19 vaccines. Finally, I would like to propose and and argue that combining innate immune memory with classical adaptive immune memory that we already use in the classic vaccines could be a very important way to go further. And we started basically studies in which we want to build basically new vaccines that are based both on activation of adaptive immunity, T and B cells in the classical way, but also combining it with trained immunity inducing ligands. And we develop new technologies actually to combine anchored trained immunity ligands with anchored antigens to induce both trained immunity and specific adaptive immunity. And we are working for different ways of administration, either through intradermal, intramuscular, or intranasal protection. And hopefully in the next couple of years, we will have new vaccines platforms which can induce at the same time improved heterologous non-specific effects and also at the same time adaptive immunity as the current vaccines are doing. And we could argue that such trained immunity approaches could be a very important addition to our armamentarium for future pandemics. So thank you very much for your attention. And I also want to thank, of course, all our collaborators who contributed to these studies.
Thank you so much to Dr. Mihai Netea. And I would also like to thank the audience for all the questions that are coming in. Once again, you can send your questions directly to Pratik Garg in a private message, and we will collect them for the end of the panel. In our next presentation, I'm very happy to welcome Dr. and Professor Robert Gallo. And, Dr. Robert Gallo requires no introduction, but I would like to say a few words. He became world famous in 1984 when he co-discovered that HIV was the cause of AIDS. After 30 years at the NIH, he co-founded the Institute of Human Virology, the IHV, and since then, Dr. Gallo has spent much of his career trying to put an end to the raging epidemic and other viral chronic illnesses. Though best known for his co-discovery of HIV, Dr. Gallo and his team have pioneered the development of the HIV blood test. His research has also helped physicians develop HIV therapies to prolong the lives of those affected with the virus. In 2011, Dr. Gallo co-founded the Global Virus Network to position the world to rapidly respond to new or re-emerging viruses that threaten mankind, to bring together the world's leading virologists and to support the training of the next generation of medical virologists. Dr. Gallo holds only 35 honorary doctorates, and he is the two-time winner of the Lasker Prize, America's most prestigious scientific award, and has been the most referenced scientist in the world. So without further ado, I welcome Robert Gallo to share his presentation, and thank you again. Sorry, Bob, I just asked you to unmute.
Oh, I'm sorry.
There we go.
I wanted to say thank you to you for your kind words, and also thank you for using your face for mine at the beginning of the meeting. It was much better. It's nice to follow Christina and Mihai. They've become friends at a distance over the last few years. This presentation will be short, nothing like the value of the first two speakers. It'll be a bit conceptual, some of it overlapping, particularly some of the things Mihai has said, but not too much. And I will not use slides, so that should be relatively straightforward. I'd like to begin by saying how I became interested in this concept of innate immunity. I don't mean at the expense of everything else, but how I tried to pay attention to it and try to see where, if possible, it may be helpful. And I'll come back to that at the very end of my comments. I was influenced by the people here, that is Mihai and Christine, Peter Abi, also who's not here, Konstantin Chumakov, who's a friend at FDA, and his long experience with polio and his strong arguments about OPV helping in protecting against influenza disease. I don't know if virus was ever mentioned. Then when the pandemic occurred, I began to think about it more and more, that I know Christine is very cautious about saying whether we could go right forward and use it for pandemics, although she sure wants to. I know that. I could, but I don't think we have to be so cautious about that. What do we have to lose? We advocated for this at the very beginning of 2020. We were received with great enthusiasm by scientists on the American committees, the so-called Warp Speed Committee, as well as NIH scientists. But interesting enough, at the last minute, it was never funded to do certain trials. You could say, well, you want to have a specific vaccine. What's the advantage of a specific vaccine? The advantage is durability. These vaccines have had a lot of positive attention. I guess deservedly so. It's new. You could change messenger RNAs right away, etc. But they're not durable. And actually, in February of 2020, we openly predicted that. Not a prediction, but it was apparent to us because the spike protein of SARS coronavirus 2 is very much like you see for influenza, like HIV. It's heavily like sugar modified with a lot of what we call mannose side chains, and they select for B cells that don't mature properly in the niches of the bone marrow to become mature plasma cells that make the long-lasting antibodies. So we're stuck with vaccines that will not last more than 4 to 6 months. I don't think that's going to change. It doesn't mean we should throw away specific vaccines, of course not, now that they're developed, but it's said to have occurred with kind of record speed, but it's still 3 years. And the next one maybe 5 years or never. So what would have been the harm of using a safe, non-specific vaccine in the middle? I'm going to jump to what I was going to say for the end, in case I forget to say it. You mentioned the Global Virus Network. I think they should in some way or other be linked with WHO, knowing we're not going to get any funding, this organization, but there's 70 centers now, 71, I think, in 40 some different countries that are linked, and linked strongly when they need to be linked strongly. Not always. Sometimes we don't get back to centers for quite a while, but this is a powerful armamentarium for pandemics that could be much more utilized, and would be very willing to develop relations with WHO. And I would assume WHO would find this of importance and interest also. We try to collect the most valuable virologists in the world, and I think we are getting there, and it happened rather fast. So especially these last few years, and now it's developing further. And I might also jump to the conclusion that I can tell you that most GVN center directors that we talk to each other, we're taking MMR. In the United States, we can't get BCG. I took MMR with my wife several times before and during the pandemic. So I did get COVID, and I was sick, and at my age, that's to be predicted, but I didn't die, and my wife did really well. So that's superiority of the women again. But anyway, let me get back to the order that I wanted to say before I talk too long rather than short. Other than being influenced by people like people here, as already said, I was thinking about this innate immunity, maybe because I read a book on bats, and bats are protected against disease from coronaviruses. And what do they have that is in common that seems to be protective is they have modest levels of chronically available interferon alpha. There's certainly going to be more things found, but not high levels. High levels are toxic to the immune system. We've got a lot of information on this and mechanisms of it that we'll be submitting for publication shortly, but at modest levels, as well as NK cells with the receptors that are for killing rather than inhibiting killing. And bats run around with that, and they're protected against coronaviruses. So I thought more about it, and then you realize I never knew this. Somebody told me, and then I look it up, and it's apparently true, that 100% of living creatures have innate immunity, but only 5% of us have adaptive immunity. And those creatures do well just with innate immunity. And I try to think of analogies to explain to people. And I thought, think of some of my ancestors trying to protect Rome. The best of them were right at the gate. I think of that as the innate immune system. And then when Christine's relatives came through the gate, we have the auxiliary troops for long-term attempt to ward off. Then you think more about it. At Mihai's ripe age, he's probably been infected 100 times, maybe more, with mild respiratory infections. Maybe he got a chill, maybe he got a low-grade fever he didn't know about, maybe he had half a day of a problem. How did he get better, folks? Do you think it's because the virus got tired or the virus ran out of cells to infect? I believe it's simply due to innate immunity. And then I started thinking more about my experience with viruses. This is not with bacteria. I don't know about bacteria, but with viruses, almost all, have at least one, and SARS has 3 genes to avoid innate immunity. Now, viruses' genomes are small, right? Why would they waste so much of their genome to avoid innate immunity? It must be, I think we can be conclusive about it, that they're scared of innate immunity, very. So if it's there in advance, it should be very interesting in warding off these infections with respiratory viruses. I mean, I don't think how conceptually you can escape that. So what if the problems are been directed, the problem of durability has really been directed by me, and he's spending his life, it seems, on trained innate immunity and how to get things to be lasting longer. Great area of need. If you can make innate immune response longer lasting and understand how to do it without toxicity and without it getting too high, this is a home run. And I always look forward to reading the next thing he might say. But he gives away his secrets slowly and doesn't tell us in advance, so I don't know what's coming next. But this is a critical area of research, quite obviously. You know, when non-specific vaccines, or whatever word we want to use, heterologous vaccines, etc., live attenuated vaccines, when should they be used? I do believe they should be used in any beginning epidemic, let alone pandemic. Why not? If you were a soldier sent to the wilds of, let's say, oh, never mind the wilds. If you were just in a big group sent, let me make an example of Egypt, and there was a new respiratory virus, not known, or no, maybe it is known, but no available specific vaccine. What would you do? Wouldn't you want to have a pocket full of cheap pills that could induce innate immunity and keep you there for a while to protect? I mean, I would and do. So when I fly to dangerous place called Europe, I always have some MMR to induce better response if I get infected. I was joking about, I'm looking at Mihai, therefore I'm making jokes always related to something like Europe right now, but forget that. So I would certainly think of that they should be used routinely at the start of an epidemic or pandemic when you don't have anything better. And later, if you don't have an effective specific vaccine. And then again, if your specific vaccine is short lasting, why wouldn't you want to do something that can be protective? And what about for poorer countries in general that can't afford or don't easily get access to the vaccines? And Jaykumar Menon's favorite subject of what about these people? And both Christine and Mihai mentioned, how about augmenting, enhancing, improving? You can enhance the adaptive immune response. In other words, making the specific vaccines better. There is a desperate need for comparison. They know that. I know that. We have to compare the differences between things like BCG, OPV, MMR, etc., which you're doing. We tried to do that. I want to say again, we got very positive response from scientists, but not higher up. It was stopped at the highest level. That's where WHO, in my mind, can be really helpful, because if they explain, get involved, people more or less, they don't have to follow, but they have to listen. And then we could do the rest. I think the comparison is one of the most important needs. And I was thinking about when you were talking about the BCG, both of you, I was thinking about, it's not a great vaccine against tuberculosis, but maybe it's a good innate immune stimulator. Well, innate immunity of the kind it's stimulating may not be terrific for tuberculosis. You're going to need a much better adaptive immune response. But for viruses, it makes sense. If you're measuring good innate immunity, particularly in my mind, NK cells and interferon, you mentioned a lot of the other cells, sure. Without dendritic cells, you don't make the interferon. So there are one. I don't know about neutrophils, basophils, eosinophils, and monocytes because we have so much less information. In Baltimore, we will start a study with Kathleen Neusel, who does delivered infections under great protection for people. She and our clinical director, Shaim Kotalil, comes from Kerala, India. Myself and a couple of others working with her. She has the Center for Vaccine Development. As you know, as it was said, I had the Institute of Human Virology, and we're almost next door to each other. So we're now talking with NIH, and things are much more positive these days. So I think this study will be done. It'll overlap with some that you previous speakers have already mentioned, but we will very likely be going ahead with this, and I hope it doesn't take forever. So that's my plan for active data. Otherwise, as I wouldn't be talking like this if I really had some information to present to you. I don't. The position of the Global Virus Network is very strong for this. So if WHO ever looked for a partner, we have already been here or there with notes that we believe this is extremely important. And as an organization and as a large number of virologists, I don't know who disagrees with it. So there's a lot of support for seeing this move forward. And the official position of our president, Christian Bruchot, who used to be president of Pasteur Institute, as well as president of INSERM, is very, very positive for this. He's probably watching this now. And as I said, we're taking our MMR when we're worried. Okay, so that's about all I wanted to say. And thanks for inviting me and letting me see colleagues.
Thank you so much, Professor Robert Gallo. And this ties in perfectly well into our last presentation with Dr. Subash Babu. Before I present our next speaker, I want to invite the WHO team in preparation. I'm going to unmute you for the panel that follows. So if you want to just raise your hand for anybody that is on the WHO team that wants to ask questions, and I welcome again the whole audience to send your questions to the chat box with Pratik Garg. So I would love to present our last but not least panelist, Dr. Subash Babu, who is a physician, scientist, and scientific director at the Indo-US Collaborative US NIH NIAID International Center for Excellence in Research, or also known as the ICER program in Chennai. His group works on the immunology, pathogenesis, and epidemiology of helminth infections, particularly that of filariasis and hookworms, TB, COVID-19, and the interaction of infectious diseases with metabolic disorders, for example, diabetes, as well as other infectious diseases like TB and helminths. The BCG and COVID-19 immunology is also part of this work, and this month he became the first Indian scientist to receive the Bailey K. Ashford Medal, which is for research in tropical medicine. Thank you so much for being here, Dr. Subash Babu, and I will pass the mic now on to you. Thank you.
Thank you so much, Alina, and thank you to the OSPF for this kind invitation to speak. I'm going to talk a little bit about some of our recent data looking at the non-specific effect of BCG vaccination that was done in elderly individuals in South India. So the objectives of our study basically were to look at two different things. One is what effect does BCG have on systemic inflammation in the elderly? And second was to look at the effect of BCG on adaptive and innate immune cell substrates. So Mihai and Kristen gave us this very nice introduction on live attenuated vaccines and their effect on innate immunity, as well as the epidemiological translational implications for that. What we were interested in was to ask the question, so what happens to elderly individuals in the first place? So elderly individuals, in terms of their immunity, there are two things that go a little awry. So first is that inflammation, baseline inflammation is increased as they age, and this is a process known as inflammaging. And the second thing is your immunity basically weakens. So both your innate and adaptive immunity weakens as you age. And so we wanted to ask if BCG had any sort of a non-specific effect on these two major processes that affect people as we age. So to do this, this was a study that was built in within a larger study looking at the effect of BCG repurposing on COVID-19 mortality and morbidity. So here we basically immunized individuals who were living in South India in COVID-19 hotspots between 60 to 80 years of age with one dose of BCG. And then a month later, collected samples from these patients and looked at a variety of inflammatory responses. So these included pro-inflammatory cytokines, chemokines, and acute phase proteins. So I'm going to be talking a lot about our data here in my presentation, and most of my data slides, or actually all of my data slides, are going to be presented in a very simple manner. So I'm going to tell you what the parameter we measured was, and what happens to the parameter post-vaccination compared to pre-vaccination. So each line represents a single individual, and you basically see whether there was any change, increase, or decrease in any of these parameters post-vaccination. And in the bottom panel of each of my data slides, I'm going to tell you what the data are in the post-vaccinated individuals compared to a group of unvaccinated controls. So when we first look at the typical pro-inflammatory type 1 cytokines, as well as the interferons that were talked about during Mihai and Christine's talk, what we find is that BCG vaccination actually dampens or significantly reduces all the type 1 inflammatory cytokines, including interferon gamma, IL-2, and TNF-alpha, decreases the IL-1 family of cytokines, as well as the type 1 interferons. And this actually holds true when we compare them with the cohort of unvaccinated individuals as well. And then when we look at other pro-inflammatory cytokines, which are much more well known because of their effect in leading to the cytokine storm as evidenced by COVID-19, such as IL-6, IL-12, IL-17, etc., we also see that BCG vaccination significantly dampens down or downregulates these pro-inflammatory cytokines as well. So you have a significant decrease in IL-6, IL-12, IL-17, GM-CSF, and a variety of other pro-inflammatory cytokines. It turns out that this effect of BCG is not confined to the typical prototypical type 1 cytokine response. BCG actually downregulates type 2 cytokines as well, and therefore appears to have an effect on potential effect in dampening type 2 inflammatory responses as well. So here we look at a variety of type 2 cytokines, including IL-4, IL-5, IL-13, all of which are significantly decreased upon BCG vaccination compared to the pre-vaccinated or unvaccinated controls. And finally, apart from the pro-inflammatory cytokines, we also see that a variety of inflammatory chemokines, which are very important for recruitment of the different innate and adaptive immune cell subsets and cause increased systemic inflammation as well as localized inflammation, are all significantly modulated, in fact, down-modulated, including a variety of CCL and CXCL chemokines upon BCG vaccination in these individuals. And much more importantly, when we actually look at the acute phase proteins, which are a direct reflection of your systemic inflammation upon any sort of vaccination or infection or any other inflammatory process, they are also significantly down-modulated. So acute phase proteins, including C-reactive protein, alpha-2 macroglobulin, and haptoglobin are all significantly decreased upon BCG vaccination compared to controls. So overall, it appears as if in terms of our first objective, when we look at what happens to systemic inflammation there is clear-cut evidence that BCG in these elderly individuals is a very significant and potent tool in down-modulating inflammatory processes that take place upon aging. We then asked what happens. So, okay, it has down-modulated a variety of markers. In fact, almost all the markers that we looked at. But really, is that the only effect of BCG? Does it not have any sort of boosting or enhancing effect on your immune system? And to answer this question, we looked at the phenotyping of a variety of innate and adaptive immune cell subsets, including T cells, B cells, dendritic cells, monocytes, and antibodies. So when we do this, what we find is that in contrast to what we see with soluble mediators of inflammation, when we look at specific subsets, here we are looking at CD4 positive T cells. And CD4 positive T cells are the memory cell compartment, the compartment that is very important and responsible for enhancing adaptive immune memory. We find that BCG actually significantly enhances the central memory as well as the effective memory compartment in the vaccinated individuals compared to the unvaccinated or the pre-vaccinated controls. And we look at CD8 T cells, the same effect is seen. So you have an increase in the frequency of central memory, effector memory, as well as the transitional effector memory CD8 positive T cell subsets. So there is significant boosting of not just CD4s, but also CD8s. And in addition, we also see a significant boosting in the frequencies of memory B cell compartments as well. So here we are looking at classical memory B cells, activated memory B cells, which are all very important for antibody production upon re-exposure, as well as plasma cells, which are the antibody-producing cells at the time of infection. And not just the memory B cell compartment is boosted, it turns out that BCG non-specifically enhances a variety of antibody isotypes. So this is looking at total antibody isotypes. This is basically completely non-specific. So we're just measuring total antibody isotypes. You look at all the IgG isotypes, G1, G2, G3, G4, all of them are enhanced by BCG. And so is IgA and IgM. So systemic levels of a variety of antibody isotypes are enhanced in these elderly individuals by BCG. And finally, if you look at innate immune cell subsets, we also find that BCG enhances the frequencies of dendritic cells as well. So we are looking at two different types of dendritic cells here, the plasmacytoid dendritic cells, as well as the myeloid dendritic cells. And both of them are significantly boosted upon BCG vaccination. So in conclusion, what I would like to tell you is basically that our studies clearly reflect two major effects of BCG, and both of which are going to have very important potential translational implications. The first is that BCG vaccination, in contrast to what you would expect of any live attenuated vaccine, does not actually increase systemic inflammation, but in contrast, it actually dampens or down-modulates systemic inflammation. And that could have a variety of important implications, including the fact that in the elderly, as we had already mentioned, inflammaging and immune senescence are two very important processes. So and when you can actually impair or dampen these processes, that actually helps in terms of boosting your immunity to protect against a variety of pathogens. And in addition to down-modulating inflammation, BCG also appears to non-specifically boost both your adaptive and innate immune cell subsets. So as we saw before, we have enhanced lymphocyte-mediated activity as well as enhanced myeloid cell responses by BCG in these elderly. So with that, I would like to acknowledge our collaborators from the National Institute of Research in Tuberculosis, as well as my group in ICER, as well as our collaborators in the NIH. Thank you very much.
Thank you very much, Dr. Subash Babu. This has been such an incredible opportunity to hear every panelist's most recent research and the evidence that has been put forward. We're also really keen to learn more about the thoughts that have been growing since all of this research has been done. I would like again to welcome the audience to ask any questions and to put those into the chat box as Pratik will be collecting them. And I also welcome at this time Archana from the WHO team and the rest of the WHO team, if you would like to come and share any questions that you may have for our dear and incredible panelists. Thank you. Archana, you are muted, but in preparation, maybe Jaykumar, would you like to expand a little bit while Archana unmutes and joins us?
Well, I thought I might just
Give a conclusion
summarize
as well.
a bit what we heard. We just heard from 4 extraordinary scientists who are working really in service of humanity. Mihai Netea, Christine Stabell-Benn, Subash Babu, and of course the legendary Robert Gallo. And what we heard is something very profound. We always think of a vaccine as protecting against one disease, or even just some variants of one disease. But we heard about vaccines that could prevent against many diseases at once, and were also ultra low cost, and that could be deployed earlier in a pandemic, and that could be available to all people and have the IP open to all. So this is really a new paradigm. It's a new scientific paradigm, and it's a new legal economic paradigm for vaccines. So I just wanted to kind of denote the significance of what we just heard. Archana, if you have comments understood, great. And if not, I understand you're in an organization with a lot of structure. So if you don't have things to say, that's fine. And then I also do have a bit of a report from Dr. Soumya to mention.
Thanks, Jaykumar and Alina. Yes, it's very inspiring and interesting to hear the discussion. No more comments from my side, please. Thank you.
Okay. Super. As a lawyer involved in international diplomacy, I fully appreciate your comment in with all of its resonance. Thank you. We did hear by text from Dr. Soumya that she would like to have further discussions on this topic with our panelists and arrange a meeting with WHO, specifically the SAGE group, which is the advisory group on vaccination strategy. So we've already had a nice positive outcome. I think we may turn it over to the group to see if folks have any questions for the panelists. And if not, we'll have some questions from this side.
All right. So again, if there's any questions or thoughts, I would like for everybody to share them in the chat. But we did have one from Eldhoth Chaco, who essentially, I think, wants to revisit Mihai's research, and as a biotech student would like to know, is this like saving a specific bookmark in DNA? And if you can revisit that part of your presentation and expand on it, so that specific part of the DNA can remember and act specifically actively against a disease.
Sure, of course. So what is basically happening is that in a normal cell, when somebody does not have an infection, did not encounter recently a vaccination, the chromatin, so the DNA is packaged very tightly usually in the nucleus. So the transcription factors cannot bind to the promoters and do not induce production of proteins which are necessary for host defense, cytokines, chemokines, defensins, and so on, because they are not needed. What is happening during the infection, there are chemical changes in the core proteins of the nucleosomes from the chromatin. And these changes, these chemical changes, make that the chromatin is no longer so tightly packaged, is more loosely packaged. And then the transcription factors can bind to the proper places, induce gene transcription, and so on. And these, the specificity of the response, which proteins are synthesized, depend on the transcription factors which are activated by a certain vaccine or infection and so on. So these genes now, turns out that they are bookmarked. These chemical changes in the histones will stay much longer than only during the infection. So the next time that there is an infection, the entire process of gene transcription can take place much easier because the chromatin at this level is a little bit more open, more loosely packaged, and the transcription factors can bind much easier and much quicker to initiate gene transcription. So during the second infection, there is a better immune response, and BCG, OPV, MMR seem to induce these long-term changes in the chromatin architecture.
Thank you so much. I also welcome the panel to share their thoughts on this process as well. Bob, I think
Can
you
we ask
wanted
a
to share.
question? Can we ask a question?
Yeah,
Please,
of course.
yeah.
Let's open this conversation.
Two questions for Subash. First is, I didn't get when after BCG vaccination did you do the assays? And the second question is, why didn't you look at NK cells, the center of innate immunity, and pay more attention to interferon alpha, the key cytokine of innate immunity?
So excellent questions, both. Actually,
No, not you. I was asking.
Oh, you were asking.
Yeah.
Oh,
So
yeah.
he may not.
Sorry about that.
Yeah, he doesn't think they're so excellent, I'm afraid.
No, that is a great question, Dr. Gallo. So we looked at 1 month after vaccination. So that was the time point that all the data that I presented. But we now have more recent data that these effects last up to 6 months. So we've looked at 6 months post-vaccination in these individuals, and the same effect of boosting both innate immunity and dampening inflammation lasts for up to 6 months in our cohort of elderly.
Just a small suggestion, though. I personally, I'd be fascinated by much earlier looking, because I think of them as protecting against infection, basically.
Right.
You know, the quick reaction that says, you know, go away, you're not wanted here. And so I would really be interested to know what happened on the first 2 weeks, you know.
Yeah, that's a great.
I agree that Mihai is telling us that things last a lot longer, and maybe that won't always be true, but at least for some things it seems to be longer. But I bet the things that are, he can tell us this, but I bet what you see early is going to be a little different than what you see many months later in terms of what's stimulated. I mean, things you'd like to see stimulated later are things you showed, antibodies and specific T cells and certain types of chemokines. But that's, you know, that's not going to happen early after infection. The purpose of the innate immunity, and you're looking at inflammation. Your question is different, really, but so I don't want to be unfair. But you know, what I'd like to look at is, are you seeing any protective responses, such as the cell in the molecule I mentioned, within the first few weeks?
Yeah, great question. So the answer to that question is that's the earliest that we looked at, unfortunately, in this study was 1 month. We haven't actually looked at immune responses earlier than that.
That doesn't answer.
So we do see so the boosting effect in terms of immune responses, that is something that we are currently doing. So we are re-stimulating the PBMCs with both SARS-CoV-2.
All right. In other words, you're thinking about it. It's going forward.
Yeah.
Okay.
Yeah. So that's something that
we don't know yet.
We do see enhanced responses to, just as Mihai pointed out in this study. These elderly individuals do make enhanced T-cell responses to SARS-CoV-2 antigens as well as to TB antigens, at least 2 different antigens that we looked at so far.
Thank you.
To answer your question about NK cells, excellent question. We actually are looking at NK cells. I didn't present that data yet. So we do have, we have phenotyped the different NK cell subsets, including looking at activating and inhibitory receptors. We don't have that complete data yet, but we do see, so the preliminary data suggests that NK cell responses are boosted as well. So both the frequencies of NK cells, the number of NK cells are increased significantly upon BCG.
Thank you so much, Dr. Babu. I would also like, Christine, you have raised your hand. I think you want to share a little bit. Am I right? If so, you can unmute and share your thoughts.
Thank you. Yes, I find it encouraging to hear all these presentations, also the very positive feedback from Bob Gallo, obviously, and from the interest expressed from WHO. I am curious to hear, since we're discussing now among the panelists also, and also related to some of the questions in the chat, what are the next steps from here, from the point of view of the panelists in terms of moving towards policy? And I'm thinking at least about how can we, given that there is so much evidence for unharvested potential of non-specific effects, how can we engage funders more in this? Clearly, this is not done by a few handful of researchers running around and getting small grants. We need some kind of solid interest and investment call for proposals from the large funders, maybe particularly also to you, Bob, and to you, Jaykumar. How do we engage the funders in this area?
Bob, you're on, you're on mute. Hi, Bob, can you unmute yourself?
I was trying, Jaykumar,
There you are.
but my hand is not as swift nor as steady as yours. No, I'm steady. Yeah, I said if you were referring to me, my track record in this field is rather bad in getting funding, but I think now it's changed, you know, because I think we've been talking to NIH about this deliberate infection experiments. How to get, I think you need to do what you're doing, make noise, get WHO on back of it, make sure they understand it. They can make the big noise. I guess being more openly critical, writing things that are more critical for the lack of funding in the area. Can we get something like the Gates Foundation very active? I don't know. I have lots of years experience with them for HIV vaccine. You know, he's obviously interested in having an OPV that's safe, right? And so, and I think I very much disagree, along with a lot of other people and every polio real expert that I know, about trying to get rid of every last polio virus, as opposed to continuing vaccination and using OPV, you know, rather than dumping it in out and destroying it all. I mean, the people that I see that I think are top level in polio, live polio, are Vladimir Egel in Russia, and Eckhard Wimmer in New York, and Konstantin Chumikov. These people that I listen to, and I've thought about it and seen about it, and I think there is a bias, a strong bias that has been built against this because people have taken a position. I understand that, you know, we all do. You take a position, you think something is right, but maybe the heels are dug in too deeply, and it's time to be gently remove the heels from the, you know, and be supportive to these notions of using non-specific vaccines, and including, and maybe even in particular, OPV. I don't know that. I think the comparisons are really urgently needed, because maybe we can avoid that argument. But OPV has great value, as you all know, because it's relatively cheap and could be made in large amounts and is something of use in developing nations. So a big killer on this is just stopping OPV. That's a harm to the field. And it's also maybe harmful to the survival of infants, if I read things properly. And it's understandable. We have a mechanism. It's not something that you're pulling out of a hat box. It's, you know, it's gaining understanding. There's science to it. And, you know, I believe, I mean, I could be led differently if somebody teaches me more about it, but I don't understand this policy. And it's a policy that was put in effect in more than one place, right? I mean, it's a, I don't, you know, I'm told it has been a CDC policy and sometimes WHO policy. I think getting that changed is important, and having discussions about it that are open and honest, not anybody putting themselves behind a position, and listen to both sides of anything on this. And, you know, if this is wrong, I would like to be, you know, corrected in an opinion. or stop expressing a view that's different. And I think that's the way we have to make progress. But we have to make noise and start stimulating discussions, and even if it's argumentative debate. I don't know what else can be done. I think that through philanthropy, you might make a breakthrough. But I believe the next years at NIH, at NIID, there'll be more interest in this area. I think that's going to happen. Other than that, we see whatever way you can continue to make noise and pressure. It's not going to happen. Yeah.
Thank
Well,
you.
you had
Thank
it.
you so much, Bob.
She asked you too, though. You were asked also, Jaykumar. You don't get off so easy.
That's
Yeah,
right. We
and
put you
then
on the hot seat. Yeah.
And then we have one more question, and then we'll close. So Jaykumar, please do share your thoughts, and we'll go to our last question.
Okay, and we may actually, I wonder if we've got 3 minutes left. So
We might go for a little bit.
okay. I think the science is there. It's indisputable that this field is worthy of more exploration. There's no doubt about it. 100 years ago, we discovered BCG had broad spectrum effects. WHO has pronounced that several of these live attenuated vaccines have multiple disease effects. The barrier is non-scientific, really. And so this calls for advocacy, it calls for networking. It's partly due to the market failure. There's no industry to take forward ultra low cost existing vaccines. So it's going to have to be creative. We'll get funding to the extent we can from the traditional sources, but we'll explore non-traditional sources, global South governments, philanthropies, individuals. The polio vaccine, the soft polio vaccine was crowdfunded. We may need to do some of this because it's an uphill battle. But what has happened over the last months is that the scientific case has become ever stronger and the mass media attention has arrived. And at Christine's Optimmunize conference, the keynotes were the WHO chief scientist and Stanley Plotkin, the dean of vaccinology. We have Denise Faustman studying from Harvard now, showing incredible effects on COVID of BCG. So the momentum is building, and we at least at OSPF will do everything in our power, and we're going to win. Simple as that.
I don't completely agree with you about industry, though, Jaykumar, because Sanofi Pasteur was ready to give us all the OPV we needed if we were doing any trial, and they were eager to find out if this would be acceptable. And what they were worried about is the political non-acceptance.
Yes, Bob, I should distinguish between the sort of industry, the generic wing of the industry that's making the low-cost vaccines and the novel vaccines. That's a good point. Yeah. Alina, what should we do? Another question or wrap up? We'll leave it in your hands.
So actually, Christine very kindly answered the last question I wanted to share, which was from Ina. And well, Christine, do you want to share a little bit more? But otherwise, there is research on boys, been fit in the beginning and girls later. So that research is available. So instead, I will close this session and thank you all in order to be courteous to everyone's time.
Let me make one point, actually, if I could.
And let Jaykumar make one last point.
And then we'll turn it to you, which is, we heard of dispatch today from the cutting edge, a new scientific paradigm and a new legal economic paradigm for vaccines. We covered the history, mechanism, and basic science, and even recent phase 3 trial, clinical trial evidence. We've made a call for 3 things: for additional studies of the old vaccines, for building novel vaccines that elicit trained innate immunity, and for certain policy changes. And I would say that the main takeaways here are 2. One, that there's cutting edge science, an alternate approach to immunity. And 2, the enormous health potential. If we could use existing vaccines and innate immunity and alternate conceptions of immunity to curb vaccine pandemics and other diseases far earlier, that simply put would change world history. It could avert trillions in economic loss and save, as we've heard today, the lives of millions of children. So I think we all on this panel collectively call on WHO, for other groups, including those on this call, to help refocus the world's strategic thinking and to help sail the global wellness ship into healthier waters. You're all present at the creation of something potentially very, very big, and we thank you very much for your time.
When are you running for president?
Thank you so much for all of for the panel's presence, for the WHO Chief Scientist, Dr. Soumya Swaminathan, and her team for being here and listening to how this shift is, it hasn't just started, it is on its way. And we encourage not only the WHO, but the whole audience to really integrate and participate in this mission, which can save millions of lives and will create the start of something very new and change the paradigm. So we thank you. I would also like, for anyone who wants to stay in touch, we will be sending you the email with this link, but you can also share your contact details here in the chat and we'll collect them so that you get more information. Don't forget to follow us to also see this webinar recording when available, and you can share that as well to, again, share the mission and share the purpose. I want to again thank our panelists, Dr. Christine Stabell-Benn, Dr. Subash Babu, Dr. Mihai Netea, Dr. Bob Gallo, as well as Jaykumar Menon, for being here and sharing so much of their research and their work. Thank you again, and until next time. My name is Alina Grenier. If you have any questions, please feel free to message us as well. Thank you.
Thank you.
Thank you.
Thank you. Thank you for organizing this.